Hi, my name is Sabrina Nicole Haverly. I work in Dr. Mishra's lab, and my project is the effects of environmental heat stress on the spleens of broiler chickens. Here we can see that chicken is used in a variety of different cuisines and cultures around the world. This is especially prevalent due to the fact that chicken is the second largest agricultural sector and the world's most consumed meat. As the world's most consumed meat, it's important that we make sure that this food source will remain a steady source of food for generations to come. The world's population is exponentially increasing, and we need to make sure that we will be able to feed these coming generations. And since chicken is on the rise in terms of popularity as a meat source, it is important to make sure that we will be able to feed this growing population chicken. Another important thing to note is that chicken is highly susceptible to heat stress. Broiler chicken specifically put on a lot of meat in a very short time frame, and this growth of meat uses a lot of energy, and this energy produces a lot of heat. When you get a lot of heat, you have a lot of stress on the body, and with chickens, it's especially important to note that chickens don't have any sweat glands. And this lack of sweat glands means that the only way they expel this internal heat is by panting, which is relatively inefficient. So if you take a bird that is already hot internally and put it in an environment that is already hot, you'll stress the bird out by decreasing the body weight, decreasing the meat quality, and it'll negatively affect the health of the bird, most importantly within the immune system. A key part of the immune system is the spleen. Now the spleen is located here, right behind the liver, and is important in things such as blood filtering, antibody production, as well as cytokine expression. Another thing to note about the chicken is that they don't have any lymph nodes. So the spleen is the primary organ when it comes to immune response. And in most cases, this works out totally fine. The chicken is able to live a healthy life. However, when it comes to stress, this puts a lot of damage onto the spleen, and it makes it very hard for the chicken to function at a normal rate. Which leads me to my hypothesis, that the immune system of the chicken will be compromised due to the heat stress imposed on the bird. For my methods, we took 48 chicks, broiler chicks, raised for meat production, and raised them up until 21 days as per the standard protocols of the Small Animal Research Facility for CTAHR. After 21 days, the chickens were then split into two groups, one with the heat stress that was raised at about 32 to 35 degrees Celsius, and chickens without heat stress that were raised at 22 to 25 degrees Celsius. After 3 weeks, the birds were then euthanized, our samples were collected, all of the weights and the relative weights were taken, and we were able to do our calculations. And certain samples were snap frozen for us to do our lab work later. For our lab work, we isolated our RNA with triazole, we synthesized our cDNA, and we analyzed all of our results with qPCR. Here for the results on the left-hand side, you can see that the relative spleen weights in the heat-stressed chickens were significantly lower than the non-heat-stressed chickens. This is especially important because it shows that the spleens were atrophied, or they stopped growing, or cells continued to die off. On the right-hand side, we can see that on the top, which is IL-12, which is a T-cell stimulating factor, which helps with cell-mediated immunity. This means that it essentially just shows us that the spleen is actually working as an immune organ to respond to the stressors within the body. On the top, we see that the heat-stress spleen is significantly higher than the non-heat stress spleen, and this shows us that the body was responding to the heat stress in the birds. On the bottom, we see CD3, which assists in activating the cytotoxic T cells and T helper cells. Now, the CD3 is essentially just going to help us get rid of the damaged cells within our body. And it is important to note that the heat stress spleen is significantly lower when it comes to the quantities as opposed to the non-heat stress spleen. And this shows us that the spleen is not able to accurately produce all of these things and fight off possible infections, and this could actually lead to death. So to conclude, due to the heat stress, the spleen was atrophied, it decreased the spleen weight, it changed the IL-12 and the CD3 expression, and it altered the health and production of the birds. Currently in our laboratory, we are investigating the detailed mechanism and mitigation strategy and trying to see if there's any dietary supplements or any other ways that we can negate all of these negative impacts on heat stress. Thank you very much.